Hi everyone. I'm going to teach you how to do streak plate technique, which is a very important microbiology technique that's used all the time in labs. Now, the purpose of streak plate technique is to isolate colonies of bacteria. So this here is a nutrient agar plate. And so you do this method that we call streak plate technique to get these circles of bacteria that you'll see. And those circles are called colonies of bacteria. Colonies of bacteria are pure cultures of that bacteria, meaning that in that little circle that you see, and you'll see it later once you've streaked the plates and the bacteria grow, those circles that you see, those colonies, are millions of bacteria of the same species. And the key word is same species. So that's why that colony is a pure culture. And so the purpose of streak plate technique is to isolate pure colonies of bacteria, and so that you're able to see the various colonies that you have growing. So we can do streak plate technique from a patient's urine sample, or maybe another body sample that you got from someone that has an infection. You can do streak plate technique from a bacterial culture that you grew in lab, or from nature, or if you want to streak something to see what types of bacteria are there. So it's a very important method. And as always, before doing this experiment, please wash your hands and clean the surface you're working on. The things you're going to need to do this experiment is one nutrient agar plate, and you can see I'm holding it agar side up, but I haven't done anything, so it doesn't matter. A Sharpie to write on your agar plate, the little sterile water that came in your bags, and then the other things that are important is you're going to need one cotton swab and 2 inoculating loops. So this is what you're going to need: a cotton swab, 2 inoculating loops, a Sharpie, the little sterile water you have, and then the nutrient agar plate. So the first thing I'm going to do before streaking my bacteria is I always like to label my agar plates. So I taught you all before that the way you label them is always start with your name. And again, as from the last video, we always, when we label agar plates, we always write on the agar side because it's very easy to lose the lid. So you always write on the back of the agar side, on the sides, not the middle, so that you could see your colonies. So I put my name, Samar, comma. I always like to put the date. Sometimes people write the date on a piece of paper, but I think it's important to put it on your agar plate. And then I always write what I'm going to inoculate on this plate, which we call the source of inoculation. So I'm going to write skin because I will be inoculating skin. So I put my name, the date, and skin on the side. And then something else I want everyone to do, we don't typically do this as microbiologists because you become so trained to do this technique, but seeing that it's your first time doing it, I'm gonna have you just draw lines on your agar plate. So just split it in half. It doesn't have to be perfect, just a line across. And then in the bottom, split that like that too. And I'm gonna label this spot as 1, 2, 3. So the top as 1, 2, 3. So the way this method works is instead of just taking your swab and just streaking it everywhere, we're trying to slowly dilute the culture so that we can see those individual colonies. So what you're going to do, and I'm going to do this, is you're going to take your cotton swab and you're going to dip it into whatever it is you're testing. Let's say you're testing a patient's urine sample, but here in this case, I'm going to do my skin. You're going to streak it across quadrant 1, and then you're going to get a new inoculating loop, take a little bit of side 1, go into side 2, then take another new inoculating loop, and then go into side 3. So I'm going to demonstrate this. So first, label your plate. Once you have your plate labeled, I'm just going to set it down. So I'm going to open my cotton swab, and I'm going to open it at the stick side with my clean hands. I have it. I'm going to dip it in sterile water. The reason I'm just dipping it in this water is just because it holds on to bacteria more. Now, obviously, if you were doing this with a patient's urine sample, for example, you're not going to dip it in water because it's already liquid. So I have water. And then what I want you to do is I'm going to go on the inside of my skin, and I'm just going to kind of be a bit rough in getting those skin cells. Okay, now I have it. And now this is what you're going to do. Make sure your plate, when you have it down, it's agar side up. I'm going to grab the plate, and so there I open the plate, and now I'm going to work with quadrant 1, and I'm going to go from the top, and I'm just going to go like this all the way to the bottom. So watch me do it. So it's going from top to bottom all the way across until I get to the middle of the plate. And then so I went down to the middle of the plate. I'm going to close my plate, and then this swab, which I'm done with, I'm just going to put it here and basically toss it in the trash. Then I'm going to open a new inoculating loop. And then also you want to open it from the stick side, not the circle, the inoculating loop side. Okay, so then grab that. And what you're going to do is take your plate again, open it. What I'm going to do is I'm going to grab a little bit from side 1 and then go down side 2. So there. Okay, and then I'm going to close it. I'm hoping that these show up in the videos. And I'm going to dump that inoculating loop, and I'm going to open a new one. The reason why you're doing a new one for each quadrant is because you're trying to dilute the culture slowly. Okay, I'm clearly having a hard time opening this, but I'm going to open it and take it out. And then what you're going to do is grab the plate, take a little bit of your bacteria from quadrant 2, and then streak quadrant 3. And that's it. That's streak plate technique. Now, and I dump everything you used in the garbage. So put it all in Ziplocs and throw it away. Here's my plate. You're going to close your agar plate. You're going to incubate it at room temperature with the agar side up. So it's going to sit on a table like this, agar side up. I anticipate it's going to take about 3 to 4 days to see growth on here. So just put it at a high surface away from children and pets where no one is going to touch it. And hopefully in a few days, you'll be able to see bacterial colonies.